I mean, sometimes it's clear. I wouldn't expect to.
You need to
you
know
would
what
test
the
that,
hormones
right?
are. Let me get into a little bit more of this glycogen stuff, and I'll try to address that a little bit more. Certainly, so far we've talked about the individual enzymes in glycolysis, and so you should certainly be able to recognize what compounds are signal active versus inhibited, and under what conditions, basically energy rich or not, do you want to run one pathway or the other? Right. In general, you don't have to remember things like if you phosphorylate glycogen phosphorylase, it becomes active. Or wait a second, does it become inhibited if you phosphorylate that particular one? Okay. Right, that would be a detail that it would probably not be reasonable for me to ask you to remember. So I would give you that if it was important. Yeah. But the big picture you should know. Let's see if we can clarify that a little bit more. So here's phosphorylase. So now we're talking about, we described, right, last couple lectures ago, or I guess on Monday, the breakdown pathway and the buildup pathway for glycogen. Actually, let's just review it. So recall that there are a set of activities to make glycogen and a set of activities to break it down. So here's the breakdown, and here's the synthesis. And the good news, or if there's any good news, is that the only part that gets regulated, so here's the breakdown. We have phosphorylase, right? We have the debranching enzyme, remember that? Then there's the mutase to take like glucose 1-phosphate to glucose 6-phosphate so we can feed it into glycolysis. In the liver, we have that mechanism through the endoplasmic reticulum. Right? The only thing that gets regulated here, well, here there's some regulation because we're bringing the G6P into the liver. But otherwise, the only thing that gets regulated is phosphorylase. And on the synthesis, remember there was that initial glycogenin protein. There's a mutase to go the other way. There's a subsidiary enzyme to make UDP glucose, the branching enzyme, and then the synthase. Right? The only thing that gets regulated here is the synthase. So right off, you kind of think, okay, there's a bunch of different steps in each side, but only two of them, one per pathway, actually are the targets of regulation. But having said that, it's very complicated on those two enzymes. Okay, so let's see how we can do it here. So phosphorylase, glycogen phosphorylase, right? So now this is breaking it down. So here's the reaction in red. Right, is active when it's phosphorylated. Right? An example of a detail that you don't have to memorize. Right? I mean, it could have gone the other way, so really, who cares? I mean, the point would be to recognize, what signals are doing this and how they're controlled. Right? So inactive and active. Right? There's a phosphatase that takes the phosphate off. There's a kinase that puts the phosphate on. These are the names of those enzymes. Again, you need to know that they're phosphatases and kinases. Do you need to remember the precise terminology of the name of that enzyme? No. Do you need to know that the kinase uses ATP? Yes. Do you need to know that phosphatase works by water hydrolysis and phosphate leaving? Yes. So here, glucagon, low blood sugar. Do you need to remember that? Yes. There's some pathway. Do you need to know the details of that pathway? No. Activates the kinase, right, which activates phosphorylase, which breaks down glucose. Does that make sense if you have low blood sugar initially? Yes. Right? So that's why glucagon is breaking this down. Epinephrine, right, is not a peptide hormone, not made from the pancreas. Instead, it's a small molecule. It looks like a benzene with some hydroxyls and another substituent on the bed. It's a small molecule compound. It is secreted by the adrenal medulla, I believe, but it's also it's secreted in the context of a fight or flight response. Right. So it is the hormone that makes you want to run away when the bear attacks. Right. Like you're afraid. Right? Or you get your final exam and you look at it and you want to run out the door. That's also happening. So I mean, we evolved in the context of the bears, but now it's turned into a final exam. So here You know, do we want to break down glucose? Yes. Where? In the muscle. Right? Why? Because we need to run away from the bear. That makes it's a hormone that operates on muscle, but it operates on this enzyme in the same way that glucagon operates for liver, because in both cases we want to break down glycogen to make glucose. In this case, in the liver, we want to break it down to make glucose so we can send it to the muscle. In the muscle, we want to break it down so we can use it for itself. Right. Calcium is another signal here that will trigger the activity because calcium is involved in muscle function. If there's low energy, AMP, then we also trigger glycogen breakdown because we need energy, so we need to go to our stores. Okay? So we have hormonal regulation, and we have obviously operating. How is it operating? By way of covalently modifying the enzyme, so that one state, the phosphorylated state, has an activity, and the dephosphorylated state does not. Questions? Okay. So here's the hormone activation. Phosphorylase B kinase. Here's phosphorylase B kinase. This slide we saw before, I think last class in fact. Glucagon, liver, hepatocyte means liver, right? Myocyte means muscle cell. Epinephrine, muscle, glucagon, hepatocyte. Right? This is the peptide, this is a little small molecule. But both of them operate through the same machinery that's in the membrane of those cells, this G protein activation machinery, which results in activation of adenyl cyclase to make cyclic AMP, which activates the kinase PKA. Okay. So PKA we saw before, last class. You can draw a little line here. It activates pyruvate kinase. When we talked about the activation of pyruvate kinase, we showed this slide. We said, oh, forget it, we'll talk about this later. For now it's pyruvate kinase, well now it's later. So what else does PKA do? It phosphorylates pyruvate kinase. It also phosphorylates a protein called phosphorylase kinase. When that's phosphorylated, it can phosphorylate glycogen phosphorylase. So here's phosphorylase kinase, right here. So that phosphorylase kinase is that phosphorylase kinase. And then we get glycogen phosphorylase and we get glucose, right? Which in the muscle is used to do the work of the muscle, and liver is used to shoot it out to the bloodstream. And the amplification cascade means you get a ton of glucose coming out from like a single binding event of a membrane. Question?
Why does it go from glycogen phosphorylase B to glycogen phosphorylase A?
Well, it's just B means inactive and A means active. That's a little confusing. Yeah, it makes it seem as though, because here phosphorylase B kinase is the same name, whether it's inactive or active. So why is this not also called the same name? It's just convention. It's like, how can we confuse you guys as much as possible kind of deal. Other questions? So this is just, so we want to have some sense of what these hormone pathways look like and how you get to the enzymes that are important, so that it's not just a total black box. Oh, glucagon, low blood sugar, boom, my enzyme. You have some sense of what's going on in between. It's a membrane process. It's a receptor. It changes conformation. A bunch of cascades. Right? I mean, you should reasonably know that there's a protein called adenyl cyclase up there. There's a PKA, which is important, not only for pyruvate kinase but also for this. So it's not unreasonable that you could remember the names of these things. Okay. All right, so we're still on glycogen phosphorylase. So we're still talking about the same enzyme regulation. So this is just showing you another level of regulation. And that level of regulation comes from the direct binding of glucose. So GLC means glucose. This is shown like this because it's a dimer. So this CH2OP